People can really relate to this from the smell of mowing their lawn.
Yeah.
And they think, oh yeah, I love that smell of fresh cut grass. Well, that's kind of a little bit sick, but it's the plants' cry for help. So the plants are screaming that you've just chopped them off.
Dr. Anjel Helms is a relatively recent addition to the Texas A&M University Department of Entomology, attaining her PhD from Penn State University in 2015. Despite being a recent hire, Dr. Helms already has over 15 publications and has investigated questions such as how chemical cues below ground can influence plants and organisms above ground. Thank you so much for joining me today.
Thank you for having me.
Yeah, absolutely. So I'd love to learn a little bit about your history, and I want to kind of get through that because I think there's a lot of really interesting things that kind of led you to where you are today. And then your research, I always call chemical ecologists or entomologists kind of the wizards of our area of research, because you all come up with all these interesting chemical concoctions and study these chemistries, and I always find it very interesting. So I'm looking forward to this discussion greatly.
Excellent.
So you started off your Bachelor's of Science in Biology with a BA in Biochemistry. You finished that in 2009 at Pepperdine University.
Mm-hmm.
And you did an honors thesis. What was kind of that work in?
So my first start into research as an undergraduate student was in plant physiological ecology.
Okay.
So in Southern California, there's a Mediterranean climate, and there are these very long stretches of drought where it pretty much only rains, I think, in maybe like January or February, and then the rest of the year, not a drop of rain. And so the plants there are very adapted to drought to be able to tolerate this. And so I was studying some of these adaptations in drought tolerance of chaparral shrub species. And more specifically, I was comparing some of the hydraulic traits or how the plants are able to transport water and while tolerating these extreme drought events with plants that were in the Santa Monica Mountains, which was close to the campus at Pepperdine, and in the mountains up around Pasadena.
So is that moving water kind of within the plant, or how they were able to locate and collect
It was
water?
how they were moving water. So taking up water from the roots and moving it up to the leaves. And one major issue with plants when they're experiencing this extreme water stress is that the xylem, which is what they use to transport the water, can become embolized. So you can have the formation of air bubbles that actually block water transport. And
Oh, wow.
so when the vessels become embolized, then they're no longer functional for the plant. And then even if the plant gets water after that, then they cannot recover from the water stress. And there are a lot of complicated complicated physics involved in explaining how the plants are able to kind of overcome this with, I guess, the various structural properties of the xylem. That was a little bit beyond what I was looking at. We were mostly just doing kind of a comparative study.
Very neat. So what was your original kind of career goal? What was your, interest at that time?
Yeah, so my original career goal, just kind of being a naive biology student who was interested in science but didn't really know what to do with it, I thought, I guess I'll just go to medical school because that's what people do. But then I got a taste of research doing this work with the plant physiological ecology, and I loved it. And so I thought, wow, this is crazy that people can build a career just studying interesting ecological phenomena. So that was really a big inspiration for me. And I did really like the work in physiological ecology, but what really transitioned my interest into chemical ecology was the plant defensive chemistry. So I noticed just how especially fragrant a lot of the plants were. Like they just produce a lot of really interesting odor compounds, and I would notice some of the plants, my professor at the time would mention, oh, these plants are really poisonous. Like don't touch your face after you've been handling this, or like avoid this plant, or avoid the poison oak. And I just really wondered, like, wow, this is crazy. Plants are making a lot of interesting things. Like, why are they doing this? And that was really then what guided my interest toward chemical ecology. And I have to say, one of the really interesting things, projects that I had a chance to get involved with as an undergraduate student was looking at the cyanogenic glycosides of a particular plant species. So
Okay.
these are molecules the plant stores. Essentially, it's an ability to produce cyanide as a toxin, obviously.
Yeah.
So the plant has attached this essentially cyanide forming molecule to a sugar, and it can store it in its tissue until the tissue becomes disrupted. So for example, by an herbivore chewing on that leaf, and when that happens, then the molecule becomes exposed to an enzyme, and then that enzyme breaks apart, like cleaves off the sugar, and then there is a rapid decomposition of the remaining unstable molecule that then produces cyanide, which is, as we know, is super toxic. And so I was studying the cyanide producing ability of one of these particular plants. It's Heteromeles arbutifolia. So we were studying that in the leaves of plants that were right around the campus in the Santa Monica Mountains, and then
Yeah.
comparing that to plants that were on the Channel Islands, so on Catalina Island. So these plants that were out on the island had, along evolutionary time, not experienced strong pressure from mammalian herbivores. And so our
Ah.
hypothesis was that they would have a reduced cyanide producing ability compared to the mainland plants. And it seemed like they did, based on our data.
Wow, very interesting. Yeah,
So
plant defense compounds is something I've just been learning more about this last year, and have really grown a huge appreciation for it. And especially just like you kind of mentioned, there's I guess a trade-off to producing some of these compounds. So if you don't have a history of herbivory, or if they're specialist herbivores versus generalists, you might expect different types of defense compounds or defense mechanisms against, your herbivores in those plants.
exactly. Yeah,
So
yeah.
that's really neat.
And so kind of that was my major motivation, was realizing that you can ask these kinds of questions, and that there's an entire field dedicated to this type of research. And I was like, wow, I'm totally sold.
So that kind of launched you directly into your PhD. Is that right? I mean, you didn't do a master's; you went straight into a PhD.
Yeah, so I did take 1 year in between my undergraduate degree and before I started my PhD, where I did a Fulbright. So I wasn't 100% sold that I was ready to go to graduate school, because when you're like 20 years old, 5 years of your life sounds like a really long time to dedicate to studying something. It's pretty laughable now. But at the time, I was like, oh my goodness, I cannot imagine doing full-time research, and this sounds intense. So my undergraduate advisor, his name was Stephen Davis at Pepperdine, and he encouraged me to go out and get some extra research experience and study. I didn't get to study abroad when I was an undergraduate, and I had really wanted to do that. And so I applied for a Fulbright fellowship to go to Germany, and one of the leading chemical ecology institutes in the world is the Max Planck Institute for Chemical Ecology in Jena, Germany. And so I was really lucky to get awarded this Fulbright to go to work in a lab in Jena under Jonathan Gershenson, and my direct supervisor was Almut Hammerbacher. And so I worked with them and got really hooked on research then, and then I started my PhD.
Wow. Yeah, that sounds like a pretty nice opportunity there. And that probably set you up with just a lot of knowledge in that area in terms of techniques and getting to know the literature and the scientists behind a lot of that chemical ecology. So that seemed like a pretty
Yeah.
pretty bright route to go.
It was pretty bright. Yeah. Yeah. So I did my PhD research under John Tooker and Mark Mesher, and I also collaborated a lot with Consuelo de Moraes. Yeah.
And so did you always, so kind of before talking about what you did during your PhD, did you always kind of want to study entomology, or was it kind of that Fulbright opportunity in Germany that got you set on chemical ecology? When was that transition? When did you realize, yeah, there's more beyond just being a doctor, perhaps?
Yeah. I definitely was not interested in studying entomology originally. It took me a little bit of time to come around to that idea. I didn't really have much of a background in insects and didn't really know a whole lot about them. And to be honest, it sounded like an incredibly nerdy thing. to study insects. And so the reason that I got into that area,
And it still is, by the way. Yeah.
it is still
Yeah,
really
yeah.
nerdy, but I'm pretty desensitized to it, I guess, now. And being one of those nerds,
Yeah, yeah.
I guess it's okay to say that. But yeah, I just thought that is so bizarre, and it seems so specialized to get a PhD in entomology. And just again, being this naive 20, like young 20s person, I was like, I'm going to go for the PhD in ecology because that seems kind of more my comfort zone. And it just so happened that some of the best chemical ecologists at Penn State, where I was really interested in working, they all were in the Department of Entomology. And so the ecology program was an interdepartmental program. And so I was in that, my degree was in ecology, but I was placed in the entomology department. And that was where I first got my taste of working with insects. And I'm very lucky that entomology departments tend to be really accepting, and just kind of convinced me basically right away that insects were fascinating and it was totally the way to go. And similar to how plants are producing so many interesting chemicals, insects are also producing a lot of chemicals, and their worlds are based around responding to chemicals. And so it's pretty much the perfect scenario for studying chemical ecology.
So, like, one of the systems that you study is the tall goldenrod and the goldenrod gallfly,
Yeah.
right? Which
Yep.
in episode 3, I talked to Dr. Brent Sinclair, and he talks about cold tolerance of the species, which can tolerate being frozen in liquid helium, right? If you treat them right, as he says, and so they have incredible cold tolerance, but also very interesting interactions with the tall goldenrod. You want to talk about that kind of briefly and what you studied there?
Sure. Yeah. So for my dissertation research at Penn State, I worked on studying the chemical ecology of this interaction with the goldenrod gallfly and the tall goldenrod Solidago altissima. And what we found is that tall goldenrod plants are able to detect or perceive the pheromone, which it's a putative sex pheromone, so it appears to be attractive to female flies. And these plants can eavesdrop on the male flies that are producing this pheromone to try to attract mates. And when they detect this pheromone, they upregulate or they prime their defenses, so that when herbivores subsequently try to feed on those plants, or when the gallflies try to lay their eggs in there and then their larvae try to feed on the plants are better defended. So the plants can chemically eavesdrop on this mating ritual of the flies and get a leg up in this coevolutionary arms race between the plants and the herbivores.
So I find this stuff kind of mind blowing, and this is what actually
I do
inspired
too.
me to go. Yeah, like, this is incredible, and this is what got me to actually even go into entomology. So I had a course with Dr. Jeremy McNeil. I don't know if you know that name at all.
He's
And
awesome. Yeah.
yeah, and so I
Gotta
took
love those bug t-shirts. He always
yeah, every
always
time.
represents the insect t-shirts.
Yeah, every lecture he wore a different bug T-shirt,
Mm-hmm.
and yeah, I took ecology under him and chemical ecology, 2 different courses, and which are the 2 courses that you are now subsequently teaching at Texas A&M.
Yeah.
Yeah,
Yeah.
and yeah, so all the examples that he gave, and how he started talking about it, especially in the context of like agriculture, like how you can use take advantage of some of these systems.
Mm-hmm.
Just kind of blew my mind and got me really interested in learning more about insects, insect ecology, and more on kind of the applied side was kind of my interest. Can you give any other kind of classic examples of chemical ecology between plants and insects?
Yeah, I think some of the really classic work has been about how when plants are damaged by an herbivore, so when a caterpillar comes along and starts feeding on the leaf and they're disrupting that tissue, the plants produce volatile compounds. So they release this pretty complex blend in a lot of cases of different terpenoid compounds, green leaf volatiles. So these are like C6, so 6-carbon alcohols, aldehydes, esters, some aromatic compounds. And so they release these odors, and a lot of people, we're really familiar with this from the smell of like fresh cut grass, for example. People can really relate to this from the smell of mowing their lawn.
Yeah.
And they think, oh yeah, I love that smell of fresh cut grass. Well, that's kind of a little bit sick, but it's the plants' cry for help. So the plants are screaming that you've just chopped them off. And so the same thing happens when caterpillars are feeding, just on a smaller scale. And the plants I mentioned that this is a warning cry. And what the plants are able to do is recruit beneficial natural enemies. So organisms at a higher trophic level, like predators, parasitoids that feed on the herbivores, they're recruited in by the plants, sort of like bodyguards, to take out those herbivores. And we call this indirect defenses. This has been studied, I guess, in probably about like the 1990s is when it first was pretty definitively shown by Jim Tomlinson and Ted Turlings, and then Consuelo de Moraes. They all kind of followed up on this work, and a lot of others. So these are just some of the names that really found that plant volatiles were very important in recruiting these herbivore natural enemies. And a really exciting aspect is that natural enemies play such an important role in agricultural systems.
Mm-hmm.
So there's been a lot of follow-up work in how can we optimize these chemically mediated interactions for better biological control. So for pest control, how can we lure in those herbivore natural enemies to help keep our plants safe?
Right, right. Yeah. I find that stuff very interesting. I also like that that way of putting it. We love the smell of suffering plants. It's
Thank
like,
you. Or when you
um,
muddle your herbs for, I don't know, making some kind of interesting thing. You're making your pesto and you're chopping up the basil and you are enjoying those aromas. Yeah, those are the plant defenses and the plant cries for help.
and so some of those examples you gave were all kind of above ground. And what's interesting about the paper we're about to discuss is you're looking at, can below ground chemical cues recruit or induce plant defenses against above ground herbivores. But before we jump into that, I want to learn a little bit more kind of about, now some of your current philosophies and or challenges you might be facing, in your relatively new faculty at Texas A&M.
Mhm.
Were there any kind of challenges, or what's it been like trying to develop and provide kind of two courses at the undergraduate and graduate level?
Yeah, it's absolutely challenging. I can't emphasize that enough. That yeah, it's a lot of work to prepare everything, and I think especially those who end up in academia and tend to be more passionate about education as well, and making sure that the students have a very quality experience.
Mm-hmm.
And I definitely can relate to that you want to do a great job of communicating the information and making sure that the students do well. And so that has been very challenging, but it's also incredibly rewarding. I thought that I liked teaching, and now I'm absolutely sure that I love it. So,
Oh, nice.
yeah.
I'd imagine as a part of what would be really rewarding is inspiring those students. Just
Mhm.
I can't tell you how much joy it brings me just to see Dr. Jeremy McNeil again, and just I have to go to him and say, hey, like thanks again, because just
Yeah.
the very fact that I'm in this whole entire field, I at least in part owe to him. And so, yeah, absolutely, you have a lot of influence over those young minds.
Yeah, it's a little scary, but yeah, no, I think that is true, and something very rewarding. So I did teach a graduate level chemical ecology class last fall,
Mhm.
and I did as part of the class. Of course, we went over a lot of getting some hands-on experience with different techniques, and then the students wrote a research proposal around some concept in chemical ecology, because that was something that I did as a graduate student, and I found it very helpful. And actually had several students in the class change a chapter or add a chapter in their dissertation around a chemical ecology topic. So that was very
Wow.
rewarding, that the students thought that it was interesting enough that they wanted to then pursue the work.
Yeah, absolutely. So now, as a research scientist, graduate student mentor, what would you say were some of the skills or life values that you think have been most important to kind of your success and where you are now in life?
Yeah, I think those who are considering going into this path, I cannot emphasize enough how important it is to get mentoring experience throughout your degree. And even if you feel like, oh, it's faster if I just do this myself, you have to learn how to delegate, and you have to learn how to mentor students, and you need to, just kind of learn how to become a manager. Because I think in a lot of people in our degree, we're very focused on learning all of the research skills. We're very focused on, okay, I need to make sure that I am the best at running this PCR reaction, or I'm the best at analyzing these samples. And that is all important, because you're going to have to teach somebody how to do that later. But it's also really important to learn how to relate to people and figure out kind of how to motivate people who are working with you. And for me, it was really challenging to learn how to kind of delegate and relinquish control. And it's like, I don't have to do everything myself now. And that's kind of a strange feeling. So I can't emphasize enough how important it is to get experience with that. And then also, you need to learn writing. So that never, I think for some people, it comes easier than for others. You need to take advantage of any opportunity to work on your writing skills and writing proposals and writing papers, and because a lot of the job is writing.
Yeah. I think that's been a common thread throughout some of the other interviews I've had as well, is writing, because you don't really think about that. I mean,
No.
You're thinking, research, you're collecting data, you're out in the field, you think about that kind of stuff, or teaching a course, teaching classes, you can think about that a little bit too, but gosh, there's a lot of writing, especially grant writing. I
Mhm.
mean, is just constant. All right. So now let's jump into one of your relatively recent research articles. So this
Mhm.
is Helms et al., published in 2019, entitled Chemical Cues Linked to Risk: Cues from Below Ground Natural Enemies, Enhance Plant Defenses, and Influence Herbivore Behavior and Performance, published in Functional Ecology. So like we had said before, there's been a lot of systems that have looked at above ground, induction of plant defenses by natural enemies or by herbivores. So what is kind of unique about this? Well, what were y'all looking at in this study?
Yeah. So in this study is some research that I started working on when I was a postdoc at Penn State with Jared Ali. And so Jared Ali comes from, his kind of background was in chemical ecology. So he started working with entomopathogenic nematodes and below ground herbivores when he was at the University of Florida, and working with Lucas Dolinsky and Hans Alborn. And so I was really interested when I saw that he was advertising for a postdoc position. I was like, you know what? Like below ground, that's where things are going on, and we just don't really know enough about that. And so I was really excited about this opportunity to look at these below ground interactions, because as you mentioned, a lot was done above ground, and I personally hadn't really thought about what was happening below ground. And another kind of interesting aspect about this work that got us thinking was, well, with plants, we often tend to compartmentalize them, and we think about the roots as a totally separate thing from the above ground portion.
Hmm.
And it's a growing field, mostly I would say probably within the last 20 years or less, that people have really started to think about how changes below ground are influencing above ground, and vice versa. So how are defenses changing kind of throughout the plant systemically? So that was kind of the basis for our thinking. And so Jared's background, as I mentioned, was with entomopathogenic nematodes. And so I wanted to do a project with nematodes. And what we realized in working with them is, so just to, I guess, to give a little more background on entomopathogenic nematodes for those who aren't familiar, they are pretty gross and pretty amazing. Um, so entomopathogenic nematodes are insect-killing nematodes. So
Like
they
tiny worms, basically.
tiny worms. Like if you look at a little dish of the free-living stage of nematodes, so we call this the infective juvenile stage. They kind of look like white lint floating in some water. So they're pretty small, but you can see them with the naked eye. So the infective juvenile stage is, as I mentioned, it's the free living stage. So they move through the soil, and there are different strategies for how they find and attack insects. So there are ambusher species that tend to, I guess, kind of hang tight and then ambush insects as they come along. And then there are also cruisers that are actively moving through the soil, looking for for their herbivore hosts. And what Jared and also some previous researchers like Sergio Rasman found was that plant roots also produce these volatile cues when herbivores are feeding on them, similar to the above ground portions of the plant, like the cut grass. So the roots are doing this too. And entomopathogenic nematodes are attracted to the odors of these herbivore damaged roots. So they're moving through the soil looking for, smelling their way, I guess, toward their insect hosts. So after they find a host, they crawl inside the body. They move in through the spiracles or through the mouth or through the anus, however they can get inside. And then they get into the hemolymph of the insect and they regurgitate a bacterial symbiont. And the bacteria, together with the nematodes, they produce this cocktail of toxins and kind of gnarly stuff. And the insect eventually dies from kind of septicemia from the bacterial infection. And then the nematodes and the bacteria, they go to work and they devour that insect from the inside out. And kind of fascinatingly, people have documented that the insects, these cadavers, I guess they're called, like these nematode infected cadavers, they're sort of preserved until the nematodes are able to consume them. They change color, so they're a characteristic color depending on which nematode and bacteria species does the infection. And then they produce these antimicrobial, antifungal, anti-scavenging compounds to preserve that cadaver for their own use. So it doesn't decompose from the soil bacteria in the same way like just a dead insect would. And what we found is that they smell really pretty terrible. These insect cadavers that are infected with the nematodes, they have
I like that you looked at that cadaver and you said, I gotta smell that and just see what it smells like. And
right? Yeah, that's kind of a sickness with chemical ecologists, is you have to kind of sniff everything.
I'm sorry, interrupted.
Yep. And so we smelled this and we thought, you know what, this doesn't smell, it smells bad, but it doesn't smell rotten. And so then, of course, I mean, in addition to sniffing something, you then have to characterize, well, what is the smell? And so we characterized the volatile cues that were being given off by those cadavers. And we found this kind of characteristic suite of volatile cues produced by the cadavers. And we thought, you know what, there's no way that the other organisms in the soil, based on what we know that's happening above ground and even in some of these below ground systems, there's no way that these odors aren't doing something, right? They must have some ecological role. And we thought, kind of had multiple hypotheses. We thought, you know what, plants are pretty incredible at their ability to detect environmental cues and to modulate their defenses in adaptive ways. So we had, from my previous research, looking at how plants could eavesdrop on insect pheromones, and knowing that plants can actively recruit these nematodes to kind of aid in their defense against herbivores, we thought, you know what, maybe plants are able to pick up on the chemical cues that are produced by these nematodes. So if it's really this characteristic, pretty potent odor source, what is the ecological significance of it? And so we had kind of competing hypotheses. We thought there were two ways that maybe plants might respond. The first way is that the plants might detect these chemical cues as a warning that, you know what, my natural enemies are present, this dead herbivore is present, that means that there's a heightened danger of herbivore damage, and so we're going to increase our defenses in the case that herbivores might come and start feeding. And that was kind of similar idea to, we thought, you know what, if you are in your house, you're sitting there and you see a lot of police on the street, what would be your response? Would you think, you know what, there's a lot of police out there, there's probably danger, I better lock my doors, I better increase my vigilance against a potential threat? Or our competing hypothesis was, you know what, maybe plants can detect the chemical cues from their natural enemies and they can relax their defenses because the bodyguards are here, they'll do their thing, we don't have to worry. So maybe you see the police on the street and you think, oh, they got my back. It's okay. I,
And
I
the
don't need to invest in
physiological
the vet.
explanation being that there's like a cost to upregulating your defenses. So why be on high alert when you can go to sleep? So it's like, yeah.
Exactly. And so we were investigating these kind of dueling hypotheses, and so we set up these experiments to expose plants to these nematode cues. And the first plant system that we started working with was potato, and we liked this system partially because they have a pretty gnarly herbivore, notorious herbivore, the Colorado potato beetle. And we thought, you know what, this is a good system to start testing this. And so we exposed potato plants to these chemical cues from the nematodes. So we had this kind of cool setup. I think there's a figure in the paper where we gently pushed some of these chemical cues from the cadavers onto the roots of the potato plants. And then we challenged the potato plants with the Colorado potato beetles. And we found that the potato plants had changed their defense responses in the systemic tissue. So in the leaves, there were heightened defenses that then reduced the performance of the Colorado potato beetle larvae that were feeding on those plants. And so we were really excited that, you know what, wow, these plants are really cueing in on this. And our findings were a little confusing because we did find sort of like a broad spectrum defense was increased, including defense responses against more of a microbial or pathogenic response.
Mm-hmm.
And so that led us to think, you know what, maybe it's not so straightforward for plants. Maybe they are a little confused, and maybe they're detecting these nematode associated cues as sort of conserved cues of nematodes. And maybe the threat of plant parasitic nematodes is so high that they're actually just fully upregulating their defenses against another threat that isn't actually present. And this is kind of how things are probably pretty likely to evolve in nature. If something is triggered for some particular purpose, and it's adaptive in some other way, an organism might be likely to continue doing this. And I guess I'm anthropomorphizing, obviously, that the plants don't have a cognitive ability to assess what exactly is that smell I'm smelling. So
Yeah. Y'all looked at two main compounds, jasmonic acid and salicylic acid. Why are those two important when we're talking about plant defenses?
yeah, great question. So the reason that we were characterizing these compounds is, so these are two plant defensive hormones. So these are signaling molecules that trigger sort of the cascade of different plant defenses. And if we didn't know specifically which defenses might be important and might be turned on, and so by measuring the different levels of these signaling hormones, we can get a broad picture of how the general defense responses might be changing. So for example, jasmonic acid regulated defenses, they tend to be associated with things like necrotrophic pathogens or herbivores that are chewing at the leaves, whereas salicylic acid defenses, and I guess the defenses that are triggered in this pathway by the salicylate pathway, they tend to be more associated with things like microbial or pathogen threats. So we can use those sort of as like broad indicators of the defensive traits.
And y'all collected those from the plant using, what's referred and is commonly used, I guess, in chemical ecologies as dynamic headspace sampling. Could you explain what that is and what makes it dynamic? I mean, is there a static headspace sampling?
There is, yeah. So we used the dynamic headspace sampling to collect the odors from the nematodes, and dynamic means that we're just pushing air over the headspace. So we have an air source that's pushing clean, filtered air over the area where the nematodes are, and then we're collecting the odors that they produce on an adsorbent filter. And if it's static headspace sampling, there are methods for this as well. It's just you're not moving air, essentially. So one technique for that would be using something called solid phase micro extraction, so SPME. And you can use these little fibers that also have this adsorbent material on them, and you expose them into the headspace, and you just let the volatile molecules attach to that fiber for some set amount of time, and then you pull it out. when you're
Oh,
done
that sounds so
collecting.
much more feasible. It sounds so
In
much
some
easier,
ways,
but
yeah, there are
okay.
trade-offs. So to doing it, it can be a little bit more difficult to quantitate the
Okay.
analysis that way. Different. This is kind of getting a little bit too into it, but some of different molecules have different affinities or different
Okay.
volatility for the binding. Whereas with the dynamic method, you're pushing air in and you're
You're
pulling it
forcing
out.
it through. It's a little
Yeah,
more
and so
active.
you
Yeah.
can. Yeah, so it's and another thing is it really depends on the system. So for plants, they do not like to be, if you're collecting plant volatiles, dynamic is definitely a good strategy because you can keep that airflow moving over the plants and then it doesn't stress them out as much. So if
Okay.
you just put a plant in
In a
a chamber
jar.
and you seal it, yeah, the plant will get
Okay.
pretty stressed.
I gotcha. And then y'all then quantified the actual, compounds coming off the plants using gas chromatography, mass spectrometry, which again is like one of those wizardry tools of chemical ecologists. Could you explain, like in very simple terms, kind of how gas chromatography works?
Sure, yeah. So essentially you put a cocktail of volatile compounds, so they're all inside of a sample. You put that into the instrument, and because of the different properties of those compounds, they get separated out over a column. So there's this really long, several meters long column that's inside of the instrument. And because of the different properties of the compounds, they bind to the column for varying amounts of time, which causes them to get separated out over time. And that's essentially what you're doing. Chromatography is just separating things. And so here we're doing it by them sticking on the inside of a long tube, essentially. And we can alter how quickly they come out of that tube based on the temperature. So we can heat things up and cause them to come out faster or slower.
And then you'd need also some standards. Is that right? Do you typically then need to, you're looking for these five particular molecules. You'll order those five molecules from somewhere that has isolated them and run in that machine as well,
Mm-hmm.
so you can compare kind of where
Yeah.
they've separated out essentially within that column.
Exactly. Yeah, yeah. You can run synthetic versions of the compounds to confirm that the timing is all correct of when they're coming off. That you're getting the correct identification of that compound.
Yeah, yeah. That's yeah. I always found gas chromatography just it's a really neat piece of tech because it's really
Mhm.
hard to imagine how can you know what molecules are in something, and so it's just a really neat piece of technology that we have as scientists to
Absolutely.
discover these things. Yeah.
Absolutely.
So you also had female Colorado potato beetles exposed to plants that were exposed to chemicals of the entomopathogenic nematodes, and they laid less clutches. I mean, the clutches will all have a similar number of eggs.
Fewer eggs. Yeah, I think
Yeah.
in total it was
Oh,
fewer eggs. Yeah.
yeah.
Yeah, that was kind of surprising to us. So we wanted to see if there was some behavioral response of the herbivores to the odors of the nematodes. So we thought, maybe it's a little far fetched that plants are going to detect these cues, but maybe these herbivores, because the nematodes are a direct threat to the herbivores, that perhaps they are responding to these cues from their predators. So there's a lot of examples in the literature of prey organisms that avoid cues from predators. So we thought, with nematodes, it probably also is happening, since we did find this reduction in the performance of the larvae on the plants, and the nematodes can directly kill the Colorado potato beetles. And so we thought, maybe they'll avoid putting their children in harm's way, and they'll uh, lay fewer eggs on these plants when we expose them to the nematode odors. And we found that was the case. There was a reduction in the number of eggs laid. And so the interesting point with Colorado potato beetle, again, is that the adult beetles are above ground and the larvae are also feeding on the leaves. But the pre-pupa and the pupa of the Colorado potato beetles are in the soil. And so in that stage, when they're getting ready to pupate, they crawl down into the soil and then they spend some time in the soil during their pupation. And so that's when they would be directly threatened by the entomopathogenic nematodes. So it's not when they're up on the foliage of the plants.
Right. And these nematodes can be commercially acquired and applied. And so in theory, so based on this, at least with potato plants, you can get both direct mortality, but also be indirectly suppressing the Colorado potato beetle by inducing the plant defenses. Is that kind of the
Hmm.
idea here?
Yeah, exactly. That was the idea, is to try to see if we can optimize our knowledge about entomopathogenic nematodes for biological control, to find if there are some additional benefits that could be gained from using them. And my lab now is following up on this work to examine in some different systems where we think that there's really high potential for using entomopathogenic nematodes in biological control. So we're working with cucurbits right now and cucumber beetle species. So in this case, the larvae are feeding for their entire duration as larvae on the roots of the plants. So they're in
Mhm.
the soil for a long time, and they're very susceptible to the nematodes. And so we're trying to see if in cases like this, where we can get direct benefits, like you said, where the nematodes are directly killing the insects, as well as inducing the plants to make them more tolerant, or more resistant, I guess, against the herbivores, whether we can get these additional benefits.
And this is called, I mean, and y'all find some potential evidence of this kind of priming of the defenses. So when they were exposed to the entomopathogenic chemical cues, in the absence of herbivores, there wasn't necessarily upregulation of salicylic acid or jasmonic acid. But then when you compared that to when there was herbivory, and either lack of or presence of those chemical cues, then all of a sudden you see an increase in better defenses, a quicker kind of upregulation of those defenses, and that's called priming. Is that right? Is that
Yeah,
yeah? Is that a common
exactly.
characteristic as well, or?
Yeah, it's pretty common. It's been observed in plants to a variety of different cues, so priming cues. And it's this kind of mysterious phenomenon where we still don't really understand the molecular mechanisms of how priming works, but we know the characteristics of what it means for the plants. So, as you mentioned, I think you mentioned before, that there is this sort of understanding that there's kind of a cost benefit ratio of inducing defense, or of, I guess, plants investing in defensive traits. So we assume that there is some cost to diverting resources to producing defenses. And plants have, what we've discovered is that plants have different layers to their ability to kind of overcome this. So we have this category of defenses called induced defenses, where the plants are not producing them, or they're producing these defenses at pretty low levels until an herbivore actually comes along and starts feeding and damaging the plant. And then the plant turns on these defensive pathways, like I mentioned, by triggering this jasmonate pathway that then, downstream of that, triggers these other defensive traits. But there are some trade-offs with inducing your defenses, and one of those is that it takes time to produce all of these things. And so there
Mhm.
can be this biosynthetic lag time in turning things on. And so plants have found sort of another strategy around this, where they can prime defenses. So if they detect a cue that is associated with an herbivore or a pathogen or some threat, but it isn't the actual event of damage, plants are somehow able to prime or get ready for the induction of defense, or this event of damage. And so, like you said, when the actual event of the herbivore feeding happens, sometime after they've been primed, they respond faster, and the response tends to be a lot stronger than in the case when they weren't exposed to those cues. And we don't really understand how it happens on the
Hmm.
molecular level,
Oh,
but
interesting.
yeah, it's pretty neat.
That could have some interesting potential implications because, in a horticultural setting, the plants arguably are not very resource limited because they are just being pumped with fertilizer. And so if you can, somehow induce those plant defenses or prime them with some kind of a chemical cue for poinsettias are always going to get white flies. So if you can prime them somehow, then you can actually at least reduce the population growth of white flies when they do get them. So it's a lot easier to manage. So it'd be really interesting to see kind of where that kind of goes
Yeah,
as we
definitely.
learn more about it.
And I think that there's a kind of an area of work around this, especially dealing with beneficial microbes. So there's been a fair amount of work with some of these beneficial plant-associated microbes that prime a sort of a broad spectrum level of resistance in plants. So plants that are associated with like these beneficial growth promoting bacteria, when they're challenged with an herbivore, they have a stronger, more resistant response.
Yeah, that's pretty neat. So
So,
what are some of the next steps in terms of some of this below ground, above ground work? Are y'all still doing some research in that area?
mhm. Yeah, so some of the students are investigating more of this kind of systemic changes in defenses. So when plants are damaged in the root by root herbivores, how is that influencing the above ground community and sort of vice versa? That's kind of an interesting ongoing field. And then I have another student that is kind of continuing on this work with the entomopathogenic nematodes.
Mhm.
And as I mentioned, investigating in squash and cucumber and in some of these different systems where we have these root feeding herbivores, looking at the responses of the cucurbit plants to this. Uh, yeah, so those are
Oh,
just a
neat.
few of the things that are
I look forward to
there.
keeping on top of some of that,
Mhm.
just out of interest sake. I think it's just really neat and has some really practical applications that can really help in an agricultural setting to at
We hope
least
so.
reduce some of the reliance, reduce reliance on, say, regular pesticide use, let's say. But anyways, I want to thank you so much, Dr. Helms, for taking the time today to talk to me about this. And I hope our listeners can appreciate a little bit more about chemical ecology.
Thank you very much.